Researchers have developed many skills and techniques over the last century in their attempt to diagnose and treat illnesses of the human mind and body. However, many illnesses continue to prove difficult in understanding what physiological mechanisms are involved. In particular, schizophrenia continues to baffle those researching it. Schizophrenia is a psychological disorder that can result in delusions and disorganized speech, thinking, and behavior. To help understand this illness, a brain stimulation technique known as transcranial direct current stimulation has re-emerged. This non-invasive technique, when applied to the scalp, sends electrodes into the brain that elicits changes in cortical excitability. This presentation will discuss the physiological mechanisms of schizophrenia and the physiological effects of tDCS, while also reviewing and critically analyzing the influence tDCS has on physiological mechanisms of schizophrenia, before finally discussing directions for future research. As mentioned earlier, the underlying mechanisms of schizophrenia have proved difficult for researchers to identify. However, it is the relationship between the neurotransmitters GABA and dopamine which have given rise to the dopamine hypothesis. As GABA is described as the main inhibitor, it can be assumed that it works to control the expression or excitation of dopamine, and thus keeps chemical balance in the brain. This is in line with the early work of Roberts, who concluded that GABA modulates dopamine by inhibiting the firing of its neurons in the substantia nigra. This was further supported by Waszak and Walters. With respect to this, the dopamine hypothesis suggests that low levels of GABA allow for high levels in dopamine. One study found that patients with high levels of dopamine exhibit symptoms of psychosis such as agitation and delusions, symptoms which are common to schizophrenia. Similarly, a study by Bell also supports the relationship between high levels of dopamine and schizophrenia-like psychosis when induced with amphetamines. Amphetamines have been found to block the reuptake of dopamine, which in turn causes an accumulation of dopamine at the postsynaptic cell. Therefore, a possible explanation for physiological mechanisms of schizophrenia may lie in the relationship between GABA and dopamine. Despite the evidence for the dopamine hypothesis, Berenson found that the blocking of glutamate receptors via administration of ketamine also causes psychosis, suggesting that other neurotransmitters may also be involved in the underlying physiology of schizophrenia. This finding of another potential cause for schizophrenia suggests the need to conduct future research into other avenues such as the glutamate hypothesis. Previously mentioned, the non-invasive technique of tDCS has regained interest amongst researchers as a tool for modulating excitability in cortical regions. tDCS facilitates the depolarization and hyperpolarization of neurons, that is, the influx of sodium ions and the efflux of potassium ions, respectively. This is achieved by applying weak electrical currents to the brain that are either positive or negative, via electrodes on the scalp. It is important to remember that tDCS does not trigger an action potential in a neuron, but instead facilitates it by boosting ongoing active neurons. Despite this, tDCS also does have its limitations, one being that it can only provide low focality due to the large size of the electrodes. However, a study by Nish et al. Found that decreasing the usual 35 cm² size of the electrode to 30.5 cm² warrants an increase in focality, but may be less effective in depth. This demonstrates the long journey researchers still have in perfecting this technique and suggests further research is required in finding the right balance between focality and effectiveness. Now that the possible physiology of schizophrenia and tDCS has been established, it is important to consider the influence that tDCS may have on the mechanisms of schizophrenia. Recent research has shown promising signs in reducing some schizophrenia symptoms when tDCS is applied. For example, a meta-analysis by Kim et al. Showed a significant reduction in the severity of auditory hallucinations when tDCS is applied twice daily. Although these findings sound promising, it is unclear what electrode placement is most efficient in relieving auditory hallucinations. This may point to the earlier idea of multiple hypotheses existing. Another study also found hyperactivation in the left cerebellum in schizophrenic patients when identifying letters amongst similar-looking letters. This increased activity in the cerebellum may reflect the visual processing abnormalities in schizophrenia, as the left cerebellum is crucial for coordinating visual motion. However, A recent study by Orlov et al. Found that tDCS reduces activation in the left cerebellum when applied. Therefore, tDCS may work in reducing hyperactivity in this region and thus reduce some of the symptoms of schizophrenia. Furthermore, tDCS capital stimulation has been suggested to increase excitation thresholds and reduce rates of neuronal firing. Therefore, this technique could be used to replicate the role of GABA as both inhibiting neuronal firing and thus would reduce the high levels of dopamine in the brain. Overall, it seems that schizophrenia is caused by an imbalance in cortical excitation inhibition, and tDCS has demonstrated potential in correcting this. In conclusion, it is evident that tDCS and the research of schizophrenia are both lacking empirical evidence to support their claims, but have shown positive potential. The directions for future research must be taken to enhance this evidence, including alternate hypotheses, improving the focality of tDCS while also maintaining its effectiveness, and simply continuing the application of tDCS on patients with schizophrenia to establish stronger evidence. However, this will only be achieved with persistent research over time. In turn, this research may be able to provide evidence for the above review and thus support the physiological effects of tDCS on the physiological mechanisms of schizophrenia.